Have you ever stopped to think about how the human brain, probably the most complex thing in the entire universe, actually builds itself? I mean, really builds itself from the ground up? It's an absolutely incredible story. It's about microscopic architects on this epic journey, a perfectly timed migration of billions of young neurons, all traveling to find their exact final spot. So let's dive into this amazing construction project.
Right, this is the big one. How does it happen? This isn't just about cells growing bigger. It's about a massive, complicated assembly job. It's all about getting the right parts to the right place at exactly the right time. And the answer to that question, believe it or not, starts with a journey. You know, before your brain could ever have a single thought, it had to move. The very cells that would one day become your memories, your ideas, your personality, they all started their lives on the go. They had to undertake this journey that was, frankly, both dangerous and absolutely essential.
Okay, so it all kicks off with this process, neurogenesis, basically the birth of new neurons. This happens deep inside the developing brain, in a special area called the ventricular zone. But here's the thing, being born is just step one. Getting to where you actually need to work, well, that's a whole other challenge.
Think of the ventricular zone like a bustling nursery where all these new brain cells are created. The problem is, this nursery is nowhere near where these neurons will eventually live and do their jobs. To build the brain's complex, layered structure, especially the all-important cerebral cortex, these brand new cells have to pack their bags and travel.
So, how do they travel? Well, the most common way is called radial migration. You can picture this as the main highway system of the developing brain. It's the most direct route from that inner nursery to the outer, developing layers of the brain.
In this highway, it isn't paved with asphalt. Oh no, it's actually made of other cells. These long, super thin structures you see here are called radial glia. They literally stretch all the way from the ventricular zone to the outer surface of the brain. They act as a kind of living scaffold, a perfect ladder for these young neurons to climb up.
So the whole process is this really simple, elegant commute. A new neuron is born, it latches onto one of these glial guide ropes, and then it just starts climbing, moving steadily outward toward its final address in what will become the cortex.
But, not all neurons take the main highway. Some are destined for different jobs, and they need to take a completely different path. You could call it the scenic route, tangential migration. So instead of moving straight out from the center, these neurons travel across the grain. They move sideways, parallel to the brain's surface, sometimes cutting across entire regions. And here's the really crucial part. They do all of this without using that radial glial scaffolding at all. They're on their own path.
And you can see the difference so clearly here. On the left, you've got that classic radial commute, with cells moving straight up. But on the right, look at those red cells. They're taking that tangential path, moving horizontally from a totally different starting point to go and populate the cortex.
So let's break it down. Radial migration is that straight shot outward, totally dependent on those glia, and it's mostly for making projection neurons, the ones that communicate between different big brain regions. Tangential migration, on the other hand, is the sideways journey. It's glia independent, and it's the main route for inner neurons, the cells that manage all the local information processing. So you've got two totally different journeys for two very different but equally important jobs.
Now this brings up a massive question, right? If you're this tiny little neuron on the move, how on earth do you know which way to go? How do you know where to turn and when to stop? Well, it turns out the brain has this amazingly sophisticated navigation system that's made of molecules.
And the source material puts it just perfectly here. These guidance cues, as they're called, act as molecular road signs. They are literally the chemical signals that create the map these migrating cells follow. Yeah, it's honestly like a biological GPS. There are attractive cues that basically scream, "Go this way," pulling neurons toward their target. And then there are repulsive cues that act as stop signs, or even as guardrails, pushing them away from the wrong neighborhoods and keeping them on the right path. And here are some of the key players in that GPS system. You've got molecules like Wnt proteins and, yes, sonic hedgehog, that's its real name, that guide cells along the big picture axes of the brain. Then you've got others like netrins, slits, and ephrins that create this incredibly complex web of come here and go away signals to fine-tune each neuron's specific path. So, what is the point of this whole elaborate, complicated journey? Well, the end goal is to build one of the most organized and powerful structures in all of nature, the 6-layered cerebral cortex. And this is the magnificent result.
This is not just some random pile of cells. It's a highly structured piece of biological architecture with 6 distinct layers. Each layer has different kinds of neurons, different connections. It's just incredible. And this level of precision is only possible because of that carefully guided migration we've been talking about.
The whole construction project follows a very specific rule, and it's known as inside-out patterning. It's a simple idea, but it's profound. The very first neurons that arrive form the deepest layers of the cortex. Then, every wave of neurons that comes after has to migrate past their older siblings to form the next layer on top.
You can see how it unfolds over time. Early on, the first arrivals come in and establish the foundation, what we call layer V. Then the next wave has to travel a bit further, past that first layer, to create layers V and 4. And finally, the last-born neurons have to make the longest journey of all, migrating all the way to the top to form those superficial layers. It's just this elegant process that builds complexity from the inside out. This entire process, from the birth of a neuron to its final placement in a specific layer, is just a marvel of biological engineering. Every single step is absolutely critical. And all of this leaves us with one final and pretty crucial question. When you have a system this complex, what happens to our health, to our brain function, when just one of these tiny cellular architects takes a wrong turn and gets lost along the way?